Is that on? Yeah, cool. So hi, everyone. Thanks so much for joining us. So before we start, I would like to acknowledge the Gadigal people of the Eora Nation, from which we're broadcasting this online seminar, and pay our respects to their elders, past, present, and emerging. So today I'd like to welcome Stephanie Chen, who is currently a PhD student at UNSW with Rich Edwards, who's joined us today, as well as with Jason Bragg at the Royal Botanic Gardens. And so Stephanie is working on genomics of various Australian plant species, and she agreed today to talk about this exciting new work she's been doing on the waratah genome. So thanks a lot, Stephanie, and off to you.
Thank you, Hervé, for the introduction and inviting me to give this seminar. So good morning, everyone. My name is Stephanie Chen. I'm a PhD candidate at the Research Centre for Ecosystem Resilience at the Australian Institute of Botanical Science and the University of New South Wales. I'm interested in developing and optimising genomic resources for the evolutionary research and conservation of Australian plants. Today, I'll be talking about assembling the waratah reference genome. Welcome to the Anthropocene, the 6th mass extinction. The International Union for Conservation of Nature lists 27% of assessed species as endangered. Much in the same vein, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services found that of the 4 million species assessed, 1 million face extinction, and that key drivers are changes in land use and sea use, direct exploitation of organisms, climate change, pollution, and invasive alien species. Specifically in the realm of plants, it's estimated 2 in 5 plant species are threatened with extinction, and the ongoing rate of plant extinctions is up to 500 times the pre-Anthropocene background rate. Some of the major threats are shown here on the proportional tree diagram. So how can genomics, the study of the complete set of genes of an organism, including structure, function, and evolution, help? Studying genomes can generate fundamental new knowledge of the organisation, evolution, functions, and interactions of organisms. This allows us to understand, utilise, and conserve biodiversity, and is facilitated by sequencing projects such as Earth Biogenome and Genomics for Australian Plants, which the waratah reference genome is a part of. With rapidly decreasing sequencing costs and technological advances, more and more plant genomes have been sequenced since Arabidopsis in 2000. However, sequencing efforts have concentrated on crops, their wild relatives, and model species. Therefore, the distribution of species with sequenced genomes across the plant tree of life is highly uneven, and there are few genomes available in the orders I'm working in. So let's have a look at the reference genomes I'm working on. So first I'll mention the Myrtaceae species from the collaborative myrtle rust research at the Research Centre for Ecosystem Resilience, Rhodamnia argentea, Syzygium oleosum, and Melaleuca quinquernovia. We are also generating references for the critically endangered Rhodamnia rubescens and Rhodomyrtus sidioides, the latter being part of BPA's Threatened Species Initiative. I've also been involved with high sea scaffolding for a UNSW venomous snake genome project Richard Edwards is leading, and these genomes are used in bioinformatics teaching. We have Tilapia speciosissima, the spectacular floral emblem of New South Wales. It was selected as one of 3 pilots for the Genomics for Australian Plants initiative, and many of you listening today may be involved with GAP projects. The reference genome individual was sampled at Mount Tomah. The polysaccharides proved difficult for DNA extractions, and it turns out that additional and younger leaf material was needed for the genome sequencing to continue as planned. However, the bushfires of 2019 to 20 swept through the area, and we thought that the plant was no longer. With much relief, we found that the individual resprouted, so presumably its lignotuber wasn't completely damaged by the intensity of the fire, and we were able to sample some new leaves. Before sequencing, the many copies of the genome are fragmented into millions of pieces. De novo genome assembly of a non-model organism is like doing a jigsaw puzzle with millions and millions of tiny pieces, and you don't have the box with the reference image. The puzzle here isn't finished, with chunks that are unanchored to the main section, and maybe there are missing pieces, like parts of a genome that were never sequenced. If this represented a draft genome, you could still calculate the size of the genome and see the main features and overall picture, which would make it fit for purpose for typical downstream applications. Genome assembly requires linkage information, evidence that allows you to fit pieces together, and different sequencing technologies serve different roles in the assembly of a reference genome. In the puzzle, areas like the sky are difficult to resolve because they're repetitive and look similar. The same goes for genome assemblies. The repetitive regions of genomes are difficult to assemble. That's where long reads are useful in comparison to short reads. Instead of 100 pieces of the same looking sky together, you have one large piece or a long read. The main issue with long reads is that they are error prone. Short reads are useful for error correcting or polishing. Linked reads also use Illumina technology, but the same barcode is attached to reads from the same long fragment, and this can provide long-range information for scaffolding. If we go back to the puzzle, linked reads would enable you to create small piles of pieces that occur in close proximity. There is also the new hi-fi sequencing, which is more accurate, and Hi-C facilitates scaffolding to chromosomes. More on that later. With the waratah, we used a nanopore long read assembly as our base, 10x linked reads for error correction, and Hi-C for scaffolding into chromosomes. So the genome size was estimated to be 891 megabase pairs using the read depth of single copy orthologs. And the species has previously been reported in the literature as a diploid with 2n equals 22. So we had 2 Promethean runs resulting in 54x coverage following base calling guppy. First, the long read nanopore data were assembled. NECAT produced the best nanopore assembly after exploring the assemblers NECAT, Fly, and Canoe, as judged by completeness, contiguity, and proximity to the estimated genome size. I did a tidy up in Diplodocus. Their haplotigs and contaminants were removed. Then there was a round of long read polishing. The 10x linked reads were then incorporated and used to correct indels and SNPs. Following this, the assembly was scaffolded and gap filled before another round of long read polishing and tidying. Let's have a look at the assemblies. Okay, so first I'll introduce you to some genome assembly terminology so we're all on the same page. So N50 is a key statistic to pay attention to, and it is the minimum contig or scaffold length needed to cover 50% of the genome. So you can have a look here. It's a measure of genome contiguity. L50 is the number of contigs whose sum length is N50. And scaffolds have gaps, so ends of unknown length, while contigs do not have gaps. BUSCO stands for Benchmarking Universal Single Copy Orthologs and is a way to assess assembly completeness. So C is complete, S is single copy, so technically each BUSCO gene should only appear once in the genome, and D is duplicated, so that could be an assembly error or an actual duplication. I used a embryo phyta set with 1,440 BUSCOs. Okay, so back to the 10x assembly. So the thing I want you to notice here is that the 10x assembly is in lots of pieces, so nearly 44,000 contigs. What can long reads do? Well, they can drastically reduce the number of contigs in the assembly, here from 11-fold to as much as 120-fold. However, if you look at the completeness, it's relatively lower due to those errors in the reads. So after polishing the NECAT long read assembly with the 10x data, things looked much better. We had 126 scaffolds with an N50 of almost 16 megabase pairs and 168 contigs with an improved BUSCO score. So we have a pretty decent draft assembly here, but we can take this to the chromosome level with Hi-C. To picture how Hi-C works, let's start by thinking about genomes in 3D space inside the cell. In each human cell, the DNA stretches out to about 2 meters long. The nucleus that houses that DNA is only 6 micrometers in size, which is about 100 times smaller than a grain of salt. The DNA cannot just get haphazardly packed into the cell. It has to remain functional for the organism to survive. So how it's packed, or the 3D conformation, is very important. For example, there are transcriptionally active, the A in yellow, and inactive, B in blue, chromosomal compartments. Hi-C is a chromosome conformation capture technique which uses proximity ligation for analysing chromatin interactions. So cells are cross-linked, resulting in links between spatially adjacent chromatin segments. Chromatin is digested with a restriction enzyme, ligation is performed, and DNA is purified and sheared. Pair-end sequencing is used to produce a genome-wide contact map. If we think about this 3D loop and representing it in 2D space, the contact map might look something like this. So much of this Hi-C research is coming from the Aiden Lab at the Centre for Genome Architecture at the Baylor College of Medicine. And his lab has illustrated how contact mapping is done with a Simpsons example, which I'm going to take you through here. So imagine you are on Homer Simpson's Facebook and you want to know how the characters interact and who lives in the same household. So we might take a look at Homer's photos. There will be many photos with his family, his wife Marge, his kids and their friends, as well as acquaintances and neighbours that he occasionally talks to. If we tally up the number of times people appear in photos together and put that into a symmetrical matrix with a heat map, it might look something like this. And you can see this darker red block here with the people he interacts most with. And that happens to be the Simpsons family. So we've used this contact map to resolve a family level interaction. The contact map can show you interactions at different scales, like if you zoom in on a map. Hi-C data is typically represented as a heat map or Hi-C contact map, where you can visualize multi-scale chromatin folding. The strength of each pixel indicates the relative pairwise contact probability of two loci. So I'm going to show you an example of Hi-C scaffolding in action in a program called Juicebox. So it's a bit like a puzzle. So if you notice here, the green represents the scaffold in the draft assembly, and the blue represents a super scaffold or a chromosome. And here we're correcting misjoins and moving the scaffold into the right place and orientation, trying to create that strong diagonal signal, bright red down the diagonal. Okay, so we have all our chromosomes now. Okay, so in the assembly pipeline, high-seq data was added to scaffold the genome into chromosomes using JUICER, the 3D DNA pipeline, and visualized in JuiceBox. So how did the waratah genome look? So here's the visualization in JuiceBox. You will notice some issues in this contact map, and after I fix those, the assembly had that strong diagonal signal. We obtained 11 chromosomes as expected, accounting for 94% of the assembly. With high-seq, our N50 has improved significantly to over 69 megabase pairs, and the completeness has also improved. Almost 40,000 protein coding genes were annotated with Genoma, a homology-based gene prediction program with evidence from the listed genomes there. So macadamia, sacred lotus, which is in the same order as waratah, Arabidopsis, and a rose. 401 ribosomal RNA genes were predicted with BARNAP, and around 700 transfer RNAs were predicted with tRNA scan SE. With a chromosome level assembly, new insights can be gained from comparative analyses. For example, synteny between waratah and the macadamia, the most important protease crop and one of the only available genomes in the family. Here, if you look at the coloured segments, they represent conserved synteny, i.e. the ortholog is present with a particular chromosome in the other organism. Next, I'm going to focus on a particular application of the reference genome, exploring the evolutionary dynamics of Tilapia. Tilapia is an Eastern Australian genus of 5 species. Previous microsatellite data and modelling suggests the history of allopatric speciation, followed by secondary contact and hybridization. The reference genomes gives us the opportunity to use Tilapia as a model clade to understand the mechanisms of speciation when combined with whole genome sequencing of each lineage. For example, exploring how much gene flow there has been and distinguishing between ancient versus recent gene flow. And if there are parts of the genome, such as rearrangements, that are actively keeping the lineages apart. It's interesting to note that each of the Tilapia species is able to hybridize to produce fertile offspring. Many hybrids are popular choices at the plant nursery. For example, these shady ladies, which are adaptable to cooler climates and lower light levels. Tilapia speciosissima is fairly widespread on the Central Coast and adjoining mountains, occurring from the Gibraltar Range in the north to Conjola in the south. Previous work by Rosetto et al. Using simple sequence repeat loci found that there are 3 distinct genetic groups. The southern populations, here in yellow, isolated by a soil barrier, show low migration and no evidence of admixture with the other populations. Amongst the northern populations, coastal and mountain populations are connected along a skewed altitudinal gradient by genetically intermediate populations. The strong association between temperature and flowering time results in a reproductive barrier between coastal and mountain populations. We obtained a whole genome shotgun dataset comprising of 2 samples from each of the 7 lineages as displayed there on the map. So 2 species of Aloxylon, the Queensland tree waratah, and Dorrigo waratah were used as the ALT group. The Illumina NovaSeq run yielded an average of 16x coverage, ranging from 7.3 to 25x across samples. If we start by looking at some astral trees, each species forms a monophyletic group as expected, and truncata is sister to the clade containing all other Tilapia. If we have a look at the lineage level, the southern lineage, which is isolated by the soil barrier, is sister to the clade containing the other 2 lineages. We in fact generated these trees before the reference genomes was available and developed a phylogenomics workflow that could be useful if you have some short read data. So this phylogenomic workflow will get you from Illumina data to species tree, and is ideal if you don't have a reference genome, have low and or variable coverage, and are looking for something quick. So genomes are assembled with a BIS. Typically, you would optimize the value of K for a single final assembly. But here we are interested in a set of K values to obtain slightly different assemblies. Previously, we used BUSCO to assess genome completeness. Here we use it to obtain orthologs. The most complete BUSCO gene from each assembly set for a particular sample is extracted using BusComp, the BUSCO compilation and comparison tool. Multiple sequence alignments are generated for IQ tree gene trees, and these many overlapping gene trees are combined into a species tree. And the key is in the multiple assemblies BusComp step, which capitalizes on the variation of genes present across K values, as well as across samples, to maximize the recovery of informative data to hopefully make those meaningful phylogenomic inferences. I'm going to introduce you to another Illumina dataset. DartSec is a method to reduce genome complexity using restriction enzymes and has allowed for population genetic studies of thousands of markers instead of dozens. We obtained the raw data reads and mapped the sequences to the reference genome and called variants with freebase, obtaining 2,739 SNPs after filtering from 196,000. A reference genome improves the reliability of variant calls, and this enhances downstream inferences. Variants were also called from the reseq data, and we had over 24 million SNPs after filtering from almost 84 million. So the final thing I'll share with you today are results from the ongoing analyses of the DART dataset. The PCA and phylogenetic network show similar groupings and reflect what we saw previously in the lineage tree. So there's good separation of truncata and aspera. However, Mongaensis and Orades are not clearly distinguished, especially where they co-occur in Monga National Park. There is some mixing of the coastal and mountain lineages of Speciosissima. Have a look here. And just to note that in this data set, hybrids were not intentionally sampled. However, we are planning to run analyses to identify potential hybrids. The structure analysis suggests K equals 3 with truncata on its own and Mongaensis and Orades together, as seen on the previous slide, but Speciosissima and Aspera forming a group. Again, in Speciosissima, there doesn't appear to be a distinct difference between the coastal and mountain lineages, although the southern lineage showing a light pink band, which is not prevalent in the other lineages. And there may be some population level patterns to explore here. For example, this orange and maroon step pattern in Mongaensis and Orades. Treemix is a method to infer population splits and gene flow in the history of Tilapia lineages. Here, with one added migration, there is gene flow from the branch of Aspera into the outgroup Aloxylon, which was unexpected, although Tilapia and Aloxylon are known to be able to cross. On the right is the colored matrix of residuals. If the standard error is above 0, lineages are more closely related to each other than in the best fit tree and are candidates for admixture. Negative residuals indicate that a pair of lineages is less closely related than represented in the tree. If we add more migration events, an introgression from the Speciosissima mountain lineage to Orades, then Aspera to the southern lineage is suggested. So how many migration events should be in the model? Delta M suggests just 1. Now we have a chromosome level Tilapia genome, which is a valuable genomic resource to understand the evolutionary origin and dynamics of Australian flora. We have also clarified some relationships in Tilapia. Next is to examine gene flow and heterogeneity across different parts of the genome and look at demographic history, which the resequencing data will facilitate. The reference genome in conjunction with the 2 datasets is an opportunity to use Tilapia as a model clade for understanding speciation dynamics. I'd like to acknowledge my supervisors, Richard Edwards and Jason Bragg, everyone at the RBG involved in this work and the funding that has made this research possible. And thank you all for listening.
Thank you so much, Stephanie. Jason, do you want to stop the recording?